Next, we're going to look on the effect of pH on the cell growth. pH affects the activities of the enzymes, and therefore it will also affect the microbial growth rate, because the microbes, they are able to release the enzyme which is required for the biocatalytic reaction. Okay, so here shows that we need to identify, to study, to know the suitable pH, okay, for the microbes to grow. Okay, so acceptable pH for growth are typically within 1 or 2 pH units of the optimum pH. The pH range can vary based on different organisms, where if the organisms is a bacteria, pH range is around 3 to 8. If it is a yeast, so the pH will be 3 to 6. And if it is from plants, so the pH will be around 5 to 6. If the organism is from the animals, so the pH will be around 6.5 to 7.5. Okay, microbes, they have the ability to control pH inside the cell, but this requires maintenance energy. We have looked on the maintenance energy before. You can recall the previous lecture on maintenance energy, which is this maintenance energy is at the declination throughout the stationary phase. Okay, because it relates with the endogenous metabolism, where this endogenous metabolism phase, the microbes are in a stage of survival. Okay, they try to find new nutrients or any remaining nutrients, and try to adapt in order for them to live, okay, and survive, okay, and repair their damaged cells. Okay, so pH, it can change due to utilizations of substrate based on the NH4 release, H ion, NO3 consumes, okay, H hydrogen, okay, and also the production of any organic acid, amino acids, or carbon dioxide, or bases. So in order to control the pH range here, if in the system, if it is in the process, we need buffer. Okay, buffer is needed, okay, in order to make sure that the pH range can be sustained in the system or in the process. Okay, this schematic diagram, it shows the effect of pH on cell growth. We can see here with the adaptation what happened. Okay, the growing is slowly increasing, okay, with the specific growth rate, okay, up towards the optimum, okay, and slowly decreasing, okay, beyond the optimum pH. But without the adaptation, what happened is, okay, the specific growth rate is rapidly increased and rapidly decreased. Next is on the heat generation by growth. For actively growing cell, the maintenance requirement is low, and heat evolution is directly related to the growth. The heat of combustion of the substrate is equal to the sum of metabolic heat and the heat of combustion of the cellular material. Okay, it can be given by this equation, delta H for substrate over coefficient of cell concentration over substrate, YXS, equals to delta H cell of the cell plus 1 over YH. Okay. Delta HS is the heat combustion of the substrate. Delta HC is the heat combustion of cell. And 1 over YH is the metabolic heat evolved per gram of cell mass produced. The above equation in heat generation, it can be rearranged into this form of equation. From the previous equation here, it can be rearranged into new equation YH equals to YXS over delta HS minus YXS delta HC. Delta HS and delta HC can be determined from the combustion of substrate and cell. Here is the energy balance of microbial utilization of substrate. Okay. We know that in the fermentation, substrate is required for the cell to grow, and if the cell, it is a aerobic bacteria, okay, so it requires oxygen here, and then what happens is throughout the fermentation, the cell concentration will be increasing, okay, and this cell, they will undergo a respiration process which able to release carbon dioxide, and there will be NH2O. Okay. So throughout this process, there will be a releasing of heat. Okay. So in this diagram, it is mentioned as a combustion of cells, right? But it is a releasing of heat throughout the respiration of the cells in order for them to keep on produce, okay, keep on reproducing and keep on growing, right? So the simple example that can be given in order to represent the heat release is if you make a tapai, you know tapai, it's a fermentation based product, Malaysian food. Okay, if you make a tapai, then if you have this tapai, okay, it is being wrapped, covered, right? So if you feel the wrap of the tapai, you can feel the heat. Okay. That's what we call the energy, the heat release by the fermentation. Okay, so only 40 to 50% of the energy stored in the carbon substrate is converted into biological energy, which is known as ATP in bio terms, during aerobic metabolism. The remainder is released as heat upon conversion of CO2 and H2O.